So I'm going to shift gear a little bit and talk to you about RNA processing, which is an absolutely essential aspect of eukaryotic gene expression. So RNA processing begins on chromatin, where newly synthesized RNA, which I'll refer to as nascent RNA, undergoes splicing, pre-mRNA cleavage, and polyadenylation. And today I'm mostly going to talk about polyadenylation, which consists in the addition of about 200 to 250 adenines to the 3' ends of nascent RNA. Once they're released from chromatin, RNAs are exported to the cytoplasm, where they'll undergo translation and eventually get degraded. And we know very well that in the cytoplasm, poly-A tails are really important for translation as well as mRNA stability. And they get progressively shortened throughout their lifetime in the cytoplasm. However, we know comparatively very little about the function and regulation of nascent poly-A tails. And in this project, we were specifically interested in understanding whether the RNA sequence itself could modulate the length that is added onto nascent RNA. So we decided to take advantage of the many naturally occurring heterozygous variants that we all carry in our cells to compare poly-A tail lengths between the two alleles from a gene. And as you're probably all aware, this is a really nice approach because both alleles will be submitted to the same environment and experimental variations. And this type of approach has allowed to identify associations between genetic variation as well as mRNA expression and mRNA processing, including alternative splicing. But first, in order to study genetic variation and how it impacts nascent poly-A tail length, we needed a method to measure these nascent poly-A tail lengths. So we start with whole cells that we biochemically fractionate into chromatin and cytoplasm. And then we extract RNA from each of these two fractions and performed direct RNA nanopore sequencing, which allows us to estimate the length of the poly-A tail on each RNA. We applied this protocol to 12 human lymphoblastoid cell lines, which generated a data set of almost 75 million reads of chromatin-associated RNA. And these were almost evenly split between the older and the newer direct RNA sequencing chemistries. After base calling and poly-A tail length estimation, we then align reads to 2 personalized genome per cell line, which then allows us to map each read to its allele of origin. And then the first question that we asked is whether nascent poly-A tail length sometimes varies between allele. And indeed, we identified 65 genes where we see highly significant differences in poly-A tail length between alleles. So here I'm showing you one of the most significant examples. And so on the x-axis, you have the 6 different individuals that were heterozygous, and on the y-axis is the poly-A tail length. And then each dot represents one read or one RNA molecule. And you can see that RNAs that originate from allele 1 have much longer poly-A tails than those from allele 2. Now, I do want to emphasize that despite the size of our data set, we're still mostly limited to analyzing highly expressed genes. So the number of genes that have these allele-specific poly-A tail lengths is likely an underestimation. So I mentioned that in the cytoplasm, there's a connection between poly-A tail length and mRNA stability. So next we asked whether we could see something similar between nascent poly-A tail length and RNA abundance. And indeed, we found that genes that display allele-specific poly-A tail length on the x-axis are enriched for genes that also have allele-specific nascent RNA expression or abundance. And we define those as genes for which one allele takes up 60% or more of the reads. So previous studies have shown that RNA hyperadenylation can target RNAs for nuclear decay. And so in that case, we would expect for RNAs with longer tails to also be less abundant. So we asked whether there was a directionality to the connection between poly-A tail length and RNA abundance on chromatin. To do that, we calculated the difference in poly-A tail length between the more abundant and the less abundant allele. And so in the case of the example that I showed you here, we would have a negative difference, which is shown on the graph. However, when we looked at all of the genes that displayed allele-specific poly-A tail length, we found both cases, either negative or positive differences. And here I'm showing you the opposite example in RPS2, where the more abundant allele also had the longer poly-A tails. So this suggests that the connection between these two metrics is not as simple as just longer tails leading to degradation. And so there's likely a multitude of factors that are affecting nascent poly-A tail length. And among those factors are possibly other RNA processing steps, because we know from several decades of study that each RNA processing step doesn't happen in a vacuum, but that they all influence each other, and that there's extensive mechanistic and physical coupling. So we asked whether poly-A tail length variations could be associated with other RNA processing steps. And this was in part motivated by our recent observation that nascent poly-A tail lengths correlate with splicing progression on chromatin. So for all genes for which we had sufficient coverage, we compared allele-specific abundance, splicing order, which is a measure of splicing progression, as well as 3' end cleavage position and poly-A tail length. And for the majority of the genes, as you can see on this upset plot, there was a single allele-specific feature. But we did identify a number of really interesting examples that displayed 2 or more allele-specific features. And today I just want to quickly highlight 2 of those examples. So first, RPS2 is the gene that I showed you briefly, where on allele 1, we found longer poly-A tails as well as higher abundance. Now, this gene also showed some really interesting splicing differences between alleles. So on this plot, you see 5 different individuals. And the proportion of reads are colored by the splicing patterns for introns 3 to 5. And so the different colors are different possible nascent RNA isoforms. And what you can see is that on allele 1, we have a really strong accumulation of the nascent RNA isoform where intron 3 is still present. It hasn't been spliced out yet. In contrast, this isoform is almost not seen on allele 2, which is instead mostly represented by nascent RNA isoforms where intron 3 has already been spliced out. So this suggests that there's a difference in timing of splicing of intron 3 for this allele, or these 2 alleles. And we know that the SNPs that differentiate between these 2 alleles are primarily located in intron 3. So our hypothesis that we're testing through additional functional experiments is that these SNPs will change the timing of splicing of intron 3, which will lead to an accumulation of this nascent RNA isoform on allele 1. And this added time on chromatin will provide more opportunity for polyadenylation, which would lead to longer tails. The other thing I want to point out is that this gene and these SNPs had not previously been associated with allele-specific splicing patterns of mature RNA. So we think that this really highlights how studying nascent RNA can reveal new layers of genetic regulation of RNA processing. So the second example that I want to tell you about is in HLA-A. And as some of you might know, HLA genes are characterized by really high levels of polymorphisms, resulting in hundreds of different alleles. It had previously been shown that some HLA-A alleles are characterized by a more distal 3' end cleavage site, resulting in a longer 3' UTR, whereas other alleles use a more proximal site. So we compared the position of 3' end cleavage to the length of poly-A tails, where for each individual that carried at least one of those 2 alleles, we looked at the distribution of poly-A tails between reads ending at proximal and distal sites. And we found that reads that end at the more distal sites tend to have longer poly-A tails than those that end at the proximal site, suggesting a connection between the position of cleavage and then the length of the poly-A tail that is immediately added onto that RNA. We also looked at the connection between splicing and 3' end position. So here on the graph, on the x-axis, you can see the 3' end cleavage position. So on the left are those RNAs that use the more proximal site, and on the right are the alleles that use the more distal site. And then the y-axis is representing the splicing order score, which in this case indicates how frequently the last intron is spliced early, before the other introns in the transcript. And what we find is that alleles using the distal site are much more likely to also have early splicing of the last intron. So together, we see 3 different allele-specific features in HLA-A, which suggests that there may be coupling between these steps, although we can't exclude at this stage that there's an independent effect of genetic variance on each feature. So in summary, I've told you about our new data set of subcellular direct RNA nanopore sequencing, which we think is a really great resource to ask a number of other biological questions. And I'd invite you to check out our preprint, where there are a lot more splicing analyses than what I had time to tell you about today. We found that poly-A tail length is either directly or indirectly influenced by genetic variation in some genes, and that several steps of pre-mRNA processing may be co-regulated by the same genetic variants. And so with that, I just want to acknowledge members of my lab, especially Simon and Louis-Philippe, who did some of the analysis that I presented today. And I also want to thank my postdoc mentor, Sterling Churchman, where this project was initially started. And I'll acknowledge our funding sources, and thank you for your attention.